Hello, everyone. I'm Michael Goodin. I'm in the plant pathology department at the University of Kentucky. I want to start by thanking Marilyn Rusink and Tyler Sacido and Lynn Bernard for their very gracious invitation to present to you and for all their help in getting this talk arranged technically. So before I get into this issue of how plant virology can inform us about emergence of zoonotic viruses such as SARS-CoV-2, I want to give thanks to, this question is not mine. It was asked by a student group in Brazil who invited me to give a talk, the NEFIT plant pathology group at the Universidade Federal de Lavras, or UFLA in Brazil. And I thought this was a very intriguing question. And so I want to encourage graduate students and undergraduates and postdocs to keep asking those sort of out-of-the-box questions, because it gives us a time to process and think about innovative ideas. And then I want to go to this slide. It's a very stressful time in this pandemic. And we need greater exposure to green spaces to sort of relax us a bit. And that's the purpose of this slide a little bit. And it's the Hall of Mosses in Olympic National Park. But it has a greater purpose to show you that the humans are down at the very bottom of the slide, and that we are part of this sort of greater picture, which are at least as big as these trees. And we tend to lose perspective on that from time to time. And so this talk sort of addresses that type of perspective. So one of the major points I'd like to make in this talk is that when the institutions that are tasked to detect and control pandemic viral diseases are operating effectively, it appears as if they're not doing anything at all, because there's hardly ever an outbreak. And when they do, they're contained. And so outbreaks become distant memories. And there's a perception that perhaps that they're no longer, outbreaks are no longer a threat. And so in 2019, in years leading up to that, new animal diseases that can also infect humans were being discovered at a rate of about 1 every 4 months. And in October of 2019, the USAID PREDICT program, which was tasked with investigating disease jumps from animals to humans, was shut down. And this sort of signaled what the response would be should a pandemic virus emerge that was being surveyed here. And so with sort of that as background, we have to sort of consider how the public perceives what an outbreak is. And it's sort of the Hollywood version of that is depicted here in this movie. And it kind of means taking some of the greatest talent that Hollywood has and giving them this really terrible script. And, you know, the job of Hollywood is to entertain. And so this is an entertaining movie and a very exciting movie. It's not a great way to learn virology, and you wouldn't want to take your definition of outbreak from this movie. So something closer to reality, and reality indeed, is this wonderful exhibit at the Smithsonian National Museum of Natural History, Outbreak, Epidemics in a Connected World. And this is really phenomenal exhibit, and it will give you insights into viruses like Nipah and Hendra and Hanta and yellow fever and smallpox, Zika and Nile and West Nile virus and the flu. And so while it is so incredibly impressive, there's no plant virus included in this exhibit. And I'm going to tell you why that should be the case and why that's important. And one of the reasons is that a lot of these viruses, most people won't even have contact with. So it's hard to make a personal connectivity to this. Some of these viruses are no longer generationally connected, and so people don't feel they have a personal connection to it, or outbreaks in general, until there is a more dramatic outbreak, like the emergence of SARS-CoV-2 in November of 2019, declared pandemic on March 11th of this year, and interestingly, declared pandemic with just about 3 times the daily case rate of what's occurring in the United States presently. And so this concept of outbreak and emergence is front and center in everybody's minds today. But there's a very important point that needs to be made here, is that we've been here before in human history, and there are 2 proven solutions to pandemics. One's vaccines. We eradicated smallpox, are well on our way to eradicating polio, measles, hepatitis A and B, yellow fever, mumps, chickenpox, human papillomavirus, and others. And if not vaccines, then antiviral drugs that have been so effective against HIV and viruses like hepatitis C. But again, these are all human infecting viruses. And it's a very different conversation that occurs, and people tend not to think about plants in conversations about viruses. And I'm going to try to convince you that that really is a problem that we have to solve. And so why is that a problem? So we need to put that into a historical context before virology as a science existed. So let's go back to 1881. Charles Darwin has just published his last scientific book. And this is 1881, this is the golden age of germ theory. So Louis Pasteur is at his height, and he's discovering the vaccine for anthrax. Carlos Finlay is really thinking about viral vectors. Yellow fever is not known to be a virus at that time, but he's implicating insects in the transmission of viruses. And yellow fever would go on to be the first human virus isolated. C-sections and blood pressure sphygmomanometers being invented. And so there's a real revolution in germ theory and medical technologies. So a year later, in 1882, Dimitri Ivanovsky, continuing on experiments initiated by Adolphe Meyer, showed that in tobacco plants with mosaic disease, if you took sap from these plants and passed them through what's called a Chamberland filter, these porcelain filters that can exclude and filter out anything larger than the size of a bacterium to make these sterile solutions. He showed if you pass the sap through that the filtrate still retained infectivity. And again, this is the golden age of germ theory. Robert Koch is discovering the bacterium responsible for tuberculosis. But here we have now a divergence of now the demonstration of a subbacterial infectious agent. And this is really the beginning of virology as a science. So really important point to realize is that virology started with plant virology, and that's a very proud moment for our science. So if we fast forward to 2019, I can't do this so much this year, but up until this point in 2019, let's say, to go to a grocery store and to find an unsuspecting customer just trying to pick out a great cantaloupe or pineapple, and to then introduce yourself and say, I'm from a college of agriculture, I can help you with that decision. If you initiate such a conversation, what you'll find is that after a few minutes, people will start to say very commonly, I didn't know plants get viruses. And that's not in a general knowledge in the public. And that's a problem. And that's a problem because 70% of our daily caloric intake comes from seeds. the recommendations in a healthy diet is that 3/4 of our diet should come directly from plants, as fruits, vegetables, grains, and then the meat and the dairy portions of those obviously require plant-based diets. So to not know that all of this agricultural productivity of plants could be affected by viruses will keep one from being cognizant of what happens when plant viruses emerge, and they can be absolutely devastating, as happened to my Uncle Warren in Jamaica when I was a small child. He was a papaya farmer and was devastated by the emergence of papaya ring spot virus. So 14 years later, Dennis Gonzalez, then at Cornell University, generates virus-resistant transgenic papaya that is practically immune to papaya ring spot virus, and this recovers the industry in Hawaii. But this is not a technology that spread due to and my uncle was not able to reap the benefit of that because of anti-GMO sentiment. But it's an important fact that scientists and virologists work for the public good. They are trying to be there to help and protect farmers. And this is our mission, and we have to tell these stories. A very important outcome of this, though, of these transgenic papaya, is that 28 years later, after deployment, this remains some of the most robust, safest, stable, cost-effective, sustainable resistance to a plant virus ever deployed. And we had the opportunity to deploy antiviral crops in a greater scale, but we didn't. For example, in potato virus Y, PVX, and potato leaf roll virus remain 3 of the major viral pathogens on potato, even though we had engineered immunity as early as 1995. In processing tomatoes, being protected by a single gene SW5 against TOSPO viruses, this is now overcome. We could have stacked genetic and transgenic antiviral strategies to help lengthen the life of that gene and protect against viruses. If you've ever been to Brazil, you know how important maracuja or passion fruit is for juices. And we've had engineered resistance, very effective, against the major viral pathogen of passion fruit since 2005. And interestingly, in sugar beet, which is 50% of US sugar, which is mostly transgenic these days, we've never stacked in to the transgenic herbicide resistance, the virus resistance that causes rhizomania. So sort of lost opportunities in agriculture. So with that as background, what we see in terms of plant virology is revealed by the emergence of SARS-CoV-2 and COVID-19. It has revealed a widespread lack of understanding of fundamental principles of epidemiology, and principles that really could have been taught with plant-based examples. And I want to give you a few of those. And so you might ask the question, well, why don't people think about viruses in discussions with plants? And one of the reasons, I think, is that well, people think that plants and animals and humans are also very different, that they don't share these sort of common disease-causing agents like viruses. So a really great question to ask at this point is just how different are we, humans and plants? And so we could play a little game, if you will, and just try to answer A or B to this series of slides. Which is the human and which is the plant? So hopefully you got that answer correctly. Which is the virus-infected human and which is the virus-infected plant? So you probably got that one right as well. Which is the human virus vector or reservoir and which is the plant virus vector? And so it's here that some people may have not quite sure. And then we get here, which is the virus-infected human cell and which is the virus-infected plant cell? And it's really here that most people would have a really hard time differentiating plant from animal or human. And I just want to show you that A is a human cell, a nucleus, infected with influenza A virus, and in B is a plant nucleus infected with Sankas Jelinek virus. And so here we have two viruses, which are negative strand RNA viruses. They both replicate in nuclei of infected cells. They're negative strand RNA viruses. And so they're genetically similar. And at this level, they appear to be very similar. And so I'll use examples, quite a few examples from the annual review of Virology chapter and Knowable magazine for the rest of the talk. And I just want to bring attention to that, that once we get inside the cell, it's very difficult to distinguish plants from animals, often for a number of phenomena. So whether or not we're talking about genetic bottlenecks in viral populations, or how viruses manipulate host membranes, or the conservation of host factors involved in virus replication or recombination, or autophagy, very similar, if not identical, suites of proteins and factors in both plants, humans. And so that's a really important thing to stress, is that in many of these phenomena, we are more similar than different. And that idea becomes very important if we then consider one of the major goals of the outbreak exhibit at the Smithsonian, that is, by understanding how humans have caused epidemics in the past, we can change our behavior and now reduce the risks of epidemics in the future. And so the idea is that global transport and travel, living close together, changing the land, natural habitat, and food production all contribute to zoonotic virus emergence. And I want to stress one point in that goal, is that this idea that we can change our behavior. And so often human behavior changes on a generational timescale. And this is why we have these legacies of social and cultural problems that are at least 400 years old. And so if we want to initiate change faster, we have to have examples where people feel personally relevant and connected to the phenomena that we're trying to explain to them. And I will argue that plant virology really serves that purpose. And so plant virology then becomes somewhat of a model system for zoonotic virus emergence. And we've been here before in science history. So the quantitative genetics invented by Gregor Mendel using peas, those principles of inheritance, are applicable in a plant system, and they're also applicable in a human system as well. And so the beauty of the model system is that it's a simplified, more tractable system that informs the more complex system, which is humans. And so what I'd like to propose is that we think about plant virology for the rest of the talk as a model system for zoonotic virus emergence. And so the hypothesis is that if plant virology can inform human virology, then the two should have in common outbreak phenomena, sudden appearance, spillover phenomena, to transfer from wild to domesticated areas, let's say, common genetics, similar type of viruses should be found in both cases. There should be a reliance on vectors for spread and a maintenance of viruses in the wild in reservoir species. So if we're going to compare plant viruses to zoonotic viruses and find these commonalities, then we need a zoonotic reference. And I'll use Ebola, which first appeared in 1976. And in the subsequent studies of this virus, what's become very important is to know the reservoir species in the wild and domestic reservoirs. How does the virus get from the reservoir into other animals that are then either consumed as food or have contact with humans. Certainly the disease etiology, epidemiology, and pathology is essential in understanding spread and transmission, and whether it's sexual transmission or not. And so since 1976, there have been these 24 Ebola outbreaks, all of which have been contained and managed. And so we've learned a lot about how to contain and manage outbreak situations, and sort of a summary of things that have to be done in an outbreak situation. You need very rapid deployment to the outbreak area of the professionals capable of dealing with such situations. Testing, screening, isolating infected patients in camps or field hospitals, quarantining people in homes or villages, and diagnostics becomes frontline in managing the outbreak. And so this is very similar in plant viruses. Virus indexing and diagnostic labs, like my collaborator Antonia Figueira at UFLA, are really frontline in tracking the initial stages of outbreak and following it through. And if we take all of that experience in how to manage outbreaks, then what we see are these curves that we've all been talking about recently. This initial increase in the number of cases, the start of the outbreak and its establishment, the so-called flattening of the curve, and then a reduction in the number of cases to the point where then the outbreak is declared over. And this requires a cross-border sharing of information, international cooperation, and accurate diagnostic and reporting of cases. So with Ebola as a reference point, let's see if we can find in plant virology those same parallel phenomena. And I'll start with the tospoviruses and Gemini viruses in plants. So tospoviruses are extremely important. So in this case here, on vegetables. And if we just look at the virus, an example virus itself, tomato spotted wilt virus, it's a envelope ambisense negative sense RNA, single-stranded RNA virus with a tripartite genome. And by its very genetics, this plant virus is similar to hantavirus, and by a broader extension of the genetics of these viruses, it's similar to Ebola, Nipah, and these zoonotic viruses of humans. Not bats, but thrips as a vector. So this plant virus has thrips as a vector. And if we look at a number of tospoviruses, we have these, we find that they have a remarkable broad genetic diversity. And so if we put this sort of information together and then ask, well, this broad genetic diversity and the segmented genome, well, is that bad in this plant virus case? Well, it's bad for the exact same reason that segmented genomes are a problem in influenza viruses, right? And so if your viral genome are like cards in a deck, you can shuffle the decks to make new hands, new viruses, new combinations of genetic material. And this happens both in influenza and tospoviruses, which makes them very devastating viruses in humans and in agricultural as well. So another example, the Gemini viruses. These viruses have circular single-stranded DNA genomes. And we're worried about the same thing with Gemini viruses as we are with zoonotic viruses. So one would go out and collect a number of isolates and sequence them and find their genetic relatedness to establish the population structure. We would ask how many variants there are in the population, and do the variants differ in virulence? And if there's a particularly severe variant, what is the probability that it may spread or emerge in other production areas? So we will want to quarantine and use the same containment strategies to prevent spread of severe strains. And not only do we need to know that for the viruses themselves. We need to know it for the vectors, the population structure of the vectors. So in this case, whiteflies and thrips. And you can just look at this panel B and think, if each one of those whiteflies gets loaded with virus, what the explosive spread of virus can be every time the wind blows, right? And so this is the problem that we have to face. And so with sort of this population structure and genetics, we absolutely worry about emergence in very much the same way that you worry about zoonotic virus emergence. So in the case of tomato chlorotic spot virus, first emerged in Brazil and Argentina in the 1980s, and then subsequent spread into the Caribbean basin in 2014. And this virus can be maintained in wild reservoir species, and it can go into a number of different crops, tomato, pepper, and tobacco shown here. And so this is very similar in structure to having, say, a bat reservoir, and then that virus capable of going into chimpanzee, humans, and macaques, say, like Ebola would. So the parallels are the same. So the other example is in cassava diseases. And so like zoonotic viruses, plant viruses show this phenomenon of being discovered and characterized, but really remaining quiescent for a very long period of time. So cassava brown streak virus was first detected in 1936, but population density, planting density, germplasm being used, changing in climatic and environmental factors all contribute to emergence. And so there's explosive spread of cassava brown streak starting about 2004. For those of you not familiar with cassava, it's one of the most important food sources in the world to about 1.5 billion people. This is an example here in Brazil, and it's fried form, mandioca frita, as you might encounter it. But it's subsistence food to hundreds of millions of people. And it is plagued by two major viral diseases, brown streak and mosaic. And so the mosaic is another one of these Gemini viruses with these circular DNA genomes. And cassava brown streak virus is a epimavirus, which, like SARS-CoV-2, is basically a giant messenger RNA that's encapsulated, that is translated to make a polyprotein and then proteolytically processed. So you could teach very effectively the genetics of SARS-CoV-2-like viruses in a plant-based system, hands-on. And just like virus emergence for zoonotic viruses, we worry about the very same thing for these cassava viruses. So we want to know, where do both viruses occur, and can you have co-infections? What are the areas where you only have one virus and not the other? And where are areas where cassava is planted on a large scale, where these viruses don't exist? And what quarantine and monitoring systems can we put in place to protect those crops in those areas? And so this is really the goals of things like the Cassava Action Project, which are using on-farm diagnostics using nanopore next-generation sequencing technologies to do this diagnosis. And then projects like the virus-resistant cassava for Africa, which has developed transgenic, very effective transgenic antiviral plants resistant to cassava brown streak. And so our initial perception of GMOs have to be reevaluated, because really this is frontline technology in combating these really important viruses. So the next example I want to get into is addresses this idea of co-infections. There's a lot of discussions now about what COVID-19 is going to be like during flu season. So what happens when influenza virus shows up? And In terms of co-infections, plant virology has a lot to say. So the example here is Maize lethal necrosis, or MLN, and it addresses this idea of synergistic viral diseases. So this viral synergy comes from having this one virus, Maize chlorotic mottle virus, which by itself is not particularly devastating in plants. And then we have these other number of polyviruses themselves that make polyproteins. And any number of these can cause disease, but usually not to a great extent. But in a co-infection, there's a synergistic amplification of the disease severity, which can be catastrophic. And so to think about synergistic interactions, we can use plant-based examples. And this MLN follows the same sort of emergence phenomena that you would see, let's say, with SARS-CoV-2 or Ebola. This first appearance in 1974 in Peru, and then subsequent pandemic virus over subsequent years. And so the world being what it is, population density and urbanization and global travel, we really have to know with increasing sophistication where all viruses are at all times, certainly the ones that cause diseases or potential for disease in humans and plants. And so this idea of spillover and spill back from wild reservoirs into domestic or inhabited areas is a really big problem, and in both plant virology and zoonosis, right? And so preventing the next epidemic really requires knowing what's out there in increasing sophistication. And that means being able to survey and collect on broad scale, to have developed diagnostic tools to characterize these novel viruses, and to generate databases that are internationally accessible. So this data can be shared to consider outbreak potential. And then sort of, if we drive this to its sort of maximum, we'd like to know every virus and every host, certainly in terms of the ones that cause diseases in humans and plants. And increasingly, we can do this with greater and greater sophistication. And so these just happen to be Gemini viruses in Brazilian non-cultivated plants, and credit to Murilo Zubini at UFV in Viçosa, Brazil. Because we need to know this information, because there will always be another outbreak. And that means we need to really train the next generation of diagnosticians, because they are really the front lines against outbreaks. And so colleagues here, Slajana Beck in Florida and Nicole Goche at University of Kentucky are really important in this. And so what I'm trying to convince you in this talk is that the factors that drive zoonotic emergence also drive plant virus emergence, be it mixed infections, genetic variability, adaptation to host, habitat conversion, reservoirs, and cultural practices. They're really parallel systems. And so with the examples I've provided, I hope I've convinced you or stimulated to think more about that. Indeed, plant virology can inform human virology and zoonotic outbreak because it has these outbreak phenomena, spillover phenomena, common genetics of the viruses, reliance on vectors, and on reservoir species. But more important, for going forward in all of this, is we spend a lot of time talking about how different things are. And there needs to be a greater conversation about how things are more alike than different at all levels of conversation. And we can start that with viruses, particularly because it's very difficult to distinguish plant from animal within the cell. So we can drive these comparisons of similarity more than differences. And so the other thing revealed by COVID-19, at least to me, is that more people need to know about plant virus emergence. And so it was really an honor to be invited to co-edit a virus's special issue that will be coming out on plant virus emergence. And I'm doing this in partnership with Jean-Marie Brichot at TAMU. It's really phenomenal collection of manuscripts being submitted, and I look forward to having that shared with you. So in addition to using plant virology as a model system, COVID-19 has revealed other aspects of our society that we really need to address. If you are an educator, instructor, doing outreach, we really have to get greater understanding of the scientific method. So in my science outreach website, I have an exercise that uses trees as a model system to demonstrate the scientific method. I'd be happy to share that with anyone, and I encourage you to use it. It's a very user-friendly, interactive procedure. We need to tell more stories about how scientists and virologists are human, and they work in the greater good, and they don't operate in nefarious ways. They truly are human working for the greater good. If we consider whether or not a virus is alive, it gives us a great thought experiment for trying to define what is living. And if you can do it relevant to a conversation about whether or not a virus is alive, it's a really wonderful conversation to be had. And it makes us increase or reduce our pettiness and increase and show that we have to have an absolute need for empathy and thinking that there are things beyond ourselves that are part of this greater good. To give us this 2020 vision, if you will, going forward in a post-COVID-19 world that has greater empathy. And so these are all at the Green Orange Cafe. And finally, given that vaccine candidates are in various stages of clinical trials, and then we will get one, we need political leadership when these are available to lead the way in the benefit of this vaccine technology. And if not political leadership, then pop culture icons like Elvis Presley, who took the Salk vaccine in 1956, to get people encouraged, because if we do that, then soon enough, we will have SARS-CoV-2 on one or both of these lists as the way to overcome solutions to pandemics. So with that, I thank you for your attention. I want to thank many of the authors and contributors and editorial staff at Annual Review of Virology. Been a phenomenal team to work with. Colleagues in Brazil, UFLA and UFV, and the College of Agriculture, Food and Environment at the University of Kentucky, a great colleagues that have many conversations contributing to this talk, the student group Nefich at UFLA, and some of these more sociology, science meets the crossroads of society. Essays are on my website and blog at the Green Orange Cafe. So thank you very much for your attention. And I look forward to answering any questions you care to email, and I'll be happy to follow up with this discussions.